Protein folding. What's the meaning of protein folding? The four level of the protein structure tend to form the most proper folded protein structure or conformation to make the protein functional and in the least energy level or consumption. The protein are folded and held together by several forms of the molecular interactions, which include thermodynamic stability of the complex, in which the hydrophobic regions tends to be interior in the protein and hydrophilic regions tend to be on the surface of the protein. This will make the molecule of the protein in the most stable thermodynamic structure. Protein have several layers, as we mentioned before, which is important in the process of protein folding. The first level, as we said, is the sequence of amino acid. This is the primary structure. The next, secondary structure, alpha helix and beta pleated sheets. The tertiary structure is the next layer of the protein structure, in which the alpha helix or beta pleated sheets will be folded in their three-dimensional structure, and the fourth level is the quaternary, which not involved in all proteins, as we said. So folding is very important to keep the molecule of the protein functional and more thermodynamically stable. This is the structure of the protein. First, the primary structure, in which there is it's the polypeptide backbone, in which there is a peptide bond, number 1, in the lower part of the figure. Then it's twisted to form the alpha helix or beta pleated sheets, or both if it's super secondary, in which it's stabilized by hydrogen bond. This is hydrogen bond, okay? And then it formed the tertiary structure or three-dimensional structure of the protein to be functional, which contain 4 types of bonds: hydrogen bonds, disulfide bonds, ionic bonds, hydrophobic interactions. This is the bonds that we mentioned in the 4 level of the protein structure. Folding is a spontaneous process that is maintained by the hydrophobic interactions, formation of intramolecular hydrogen bonds, and electrostatic attractions. This is unfolded protein, just a polypeptide chain which is not connected. Both polar and non-polar side chains are beside together or beside the water molecule. But when it's probably folded in aqueous environment, which is our body, aqueous environment, the hydrophobic regions tend to be interior in the molecule, and the polar regions, which are the hydrophilic, tend to be outside the molecule. And this is stabilized only by hydrophobic and hydrophilic interactions, hydrogen bonds, and electrostatic attractions. Protein folding must be thermodynamically favorable. It's the least thermodynamic consumption for the protein molecule, and it's a spontaneous reaction. Minimize the number of hydrophobic side chain exposed to water or aqueous environment, and is important driving force behind the folding process. Protein denaturation. It's type of unfolding of the protein. Protein structural changes resulting in the loss of three-dimensional structure of the protein needed for its function. This is called denaturation. Without hydrolysis of the peptide bond, all bonds will be lost except the peptide bond. In the denaturated state, the conformation of the protein needed to be not to be completely randomized and can be sometimes reversed. Sometimes it's irreversible. What makes the protein denaturated? Physical factors like heat, extreme pH, and chemical factors like organic solvents like alcohol, acetone, certain solutes like urea, guanidine hydrochloride, or detergent can form protein denaturation. Denaturation often results in aggregation and precipitation of the unfolded protein. This is the normal protein. When there is factor denaturating it, like agents, heat, pH, ionic, some chemical agents, it will lose its bonds except the peptide bond and lose its biological activity and converted into denaturated protein, which may be aggregated and precipitated. Sometimes it can be reversed and renaturated again. This is the mean our definition of denaturation. In our body, normally there is what called chaperones. Chaperones or heat shock protein, HSP, are proteins that interact with the unfolded or partially folded or improbably folded polypeptide chains, facilitating it to be correctly folded. احنا زي ما قلنا it's spontaneous process. All proteins tend to be folded by the hydrophobic and hydrophilic forces. What makes it easy process? The chaperones or heat shock proteins. There are two major classes of chaperones: molecular chaperones like HSP70 family and chaperonines like HSP60 family of protein. Molecular chaperones, which is HSP70 family, help to guide the folding of the newly sensitized protein. They bind to the hydrophobic patches that are exposed or incompletely folded of the protein using repeated molecules of ATP binding and hydrolysis, which is required for the proper folding. This process is needing energy. This process needs energy, which is coming from converting of ATP into ADP and giving energy to the HSP protein machinery to make or guide the hydrophobic regions of the protein into the interior side of the protein like this, and finally make it correctly folded protein. So when the newly sensitized protein, it acts mostly on the newly sensitized protein, ATP come to be hydrolyzed into ADP, giving energy to HSP70 machine, which is the red one, and this will guide the hydrophobic regions of the newly sensitized protein in the interior side of the protein away from the surface, and the hydrophilic remain on the outer surface of the protein, and this will consume ATP then be detached from the protein, giving the correctly folded protein. This is the process of acting of the molecular chaperones on the newly sensitized protein. The second category of chaperones is the HSP60 family, which is called chaperonin, which acts on the fully sensitized protein but not have yet achieved the final folded form. The presence of incompletely folded protein can lead to formation of aggregates and dangerous consequences for the cell and can make disease. So it's very important to keep the protein properly folded in our cells. How it's the molecular chaperonin HSP60 act? It's barrel-like. Here is the incompletely or incorrectly folded protein, which is fully sensitized. Enter the barrel or the machine of HSP60 protein complex. Here is the site where the hydrophobic protein or the hydrophobic regions of the unfolded protein will be attached. This will consume also ATP, and then a cap of the machine will be closed, consuming ATP for correctly fold the protein, and finally it will expel the fully correctly folded protein at the end. Protein misfolding disease. Like we said before, unfolded protein or misfolded protein in the cell will form aggregates and will form a dangerous consequences in the cell. Many inherited diseases result from mutant proteins that evade the quality control process and fold abnormally and form aggregates. The gradual decline of the protein quality control with age can also lead to disease by permitting normal protein to form aggregates that can impair the cellular function. Many neurodegenerative diseases like Huntington and Alzheimer are associated with the presence of misfolded protein aggregates in the neuronal tissue. The aggregates in patients can be intracellular, nuclear, or cytoplasmic, or extracellular. In Alzheimer, there is a soluble protein that is normally produced in the neuronal cells, which is misfolded by the age, and the protein process machine will decline in its function, leading to misfolded state and convert this soluble protein into insoluble extracellular amyloid fiber. Commonly, fiber formation is promoted by aggregation of regions of the protein that have beta conformation, beta pleated sheets. It leads to aggregation in the beta pleated sheets. In Alzheimer, the proteolytic cleavage of the neuronal membrane protein amyloid beta precursor protein APP produce amyloid beta peptide that converted into beta amyloid aggregates and into amyloid fibrils. These amyloid fibrils which will cause Alzheimer. The extracellular deposition of the amyloid fibrils is associated with plaque formation and death of the neurons. Let's see the figure. This is the native protein. It will be misfolded. There is no correcting process of the unfolded protein. So in the beta conformation regions, which contain beta plated sheets, there will be a formation of the amyloid fibril structures, leading to precipitation and aggregation of the amyloid beta peptides and the amyloid fibrils in the beta conformation regions. These aggregates extracellularly will form plaques and leading to death of the nearby neurons. This is the principle of happening of Alzheimer's disease. Thanks, and I hope you understand, and I'm waiting for your feedback on the email or on our office hours. Thanks.